Hello, everyone. Welcome back to my video tutorial for TCR sequencing data analysis. In this video tutorial, I'm going to show you how to perform database query for your TCR sequencing dataset. So to perform database query, we need to use the publicly available database. For today's demonstration, we are going to do epitope query. There are 5 common large-scale databases with TCR epitope pairs are available. We are using Scirpy for the analysis, so at the moment, you can load the IE database and the VDJ database into Scirpy. The other 3 databases, MC TCR, PIRD, and Immune Code TM, at the moment, they are not incorporated into the Scirpy package. So for today's demonstration, we are going to use the VDJ database. This database contains the largest variety of different SARS-CoV-2 epitopes. Because the Scirpy has the database, so we can load the database as VDJ-DB. Let's load it and also have a look at the database. If we run, you can see we loaded the database as VDJ-DB. It has 62,177 antigen epitopes. You can see here is the observations, including the information for antigen epitope, antigen gene, antigen species, and also other information. We can also have a look at the database observations to see the metadata in details. You can see in the epitope antigen, here the amino acid sequence. Then here are the gene name. You can see in the antigen species column, we have the antigen species name. For example, here we can see HIV1 and also weight. There are more information in the metadata. You can have a look at them in details. So here is the metadata information. We can also have a look at the antigen epitope count for each antigen species. So in total in this database, we have 42 antigen species. If we run, you can see the highest epitope count is the CMV virus. Then the second highest count for the antigen epitope is the influenza A. You can see SARS-CoV-2 has 6,237 epitopes. So here you can see the total epitope count for each species. In total, we have 42 species here. So in this code, I showed you how to look at the epitope count for all the species. If you are interested in one of the species, for example, the HIV virus, you can just look at the count value for HIV. Let's run this one. You can see there are 3,408 count in total for HIV virus in this dataset. So this is the total count for the antigen species. We can also look at the individual count for different antigen epitope. Let's run. You can see now it's the antigen epitope count for HIV virus in this dataset. You can see this amino acid sequence has the highest count, 780. And here we have the list for all the amino acid sequence in this dataset for the HIV-1 virus. So I showed you how to load the VDJ database, and also we had a look the information for this database. So now we can load our query data to perform epitope query for our query dataset. So we are going to use the 3 key TCR demonstration data that I performed all the analysis in video 1 to video 4. So I save the data. If you don't know how to save the data, you can use the data write to save the data as a h5mil data file. Now we can just load the data from previous analysis. Let's load the data now. You can see we loaded the data back again as the data. So we have the reference data VDJ DB, and also now we loaded the query data data. So first we need to use the immunoreceptor distance function again to compute the sequence-based distance matrix for all the VJ and VDJ sequence. We used this function to analyze the TCR sequencing data for the 3K demonstration dataset. So you can see the settings here. The matrix will be identity, and the sequence will be amino acid sequence. So we want to identify the identical amino acid sequence between the query dataset and the reference dataset. So let's compute the amino acid sequence-based distance matrix. You can see it computed the sequence distance first for VJ sequence, then next for the VDJ sequence. So after the calculation, Scirpy should save the data into the metadata. We can have a look at the metadata for the TCR sequencing data. Let's run. You can see here we saved the calculation as IR distance VDJDB amino acid identity. So we have the amino acid sequence-based distance matrix. Now we can perform the query analysis to identify identical amino acid sequence in the reference dataset for each cell in our query dataset. So we can perform the query now. You can see following the analysis, it stored the IR distance matrix in the IR query VDJDB amino acid identity. We can have a look at the metadata again. You can see now we have a new column, IR query VDJDB amino acid identity. So we performed the data query analysis. Now we can transfer the annotation from the reference dataset to the query dataset. Again, the query dataset is data and the reference dataset is VDJDB. We want to identify the identical amino acid sequence. For the annotation, we want to get the information for the antigen species and also the antigen gene. Let's perform the annotation. So you can see after the annotation, we will have two metadata columns, the antigen species column and also the antigen gene column. So we should save the data, but if you want to create a data frame for the epitope query analysis we performed here, you can use this code to create a data frame, and we name it as antigen. Let's create the data frame. We can have a look at the data frame for the last 5 rows because we are using the tail function. You can also use the head function to look at the first 5 rows, or you can just use the antigen full name to look at the first 5 and the last 5 rows. Let's look at the last 5 rows. You can see here is the data information we generated for the Epitope query. You can see the row names are the cell barcode in the TCR sequencing dataset. Then we have the antigen species and the antigen gene information for the TCR clone type in each cell. If you want to save the data, you can use the write function to save it as a CSV file. Let's save it. If you have a look at my TCR folder, you can see we have a antigen CSV file saved here. Let's open the file and have a look. You can see the first column is the barcode, the second column is the antigen species, and the third column is the antigen gene. So we annotated the antigen epitope for the TCR clone type in each cell. If you want, you can also plot the epitope on UMAP cell canisters. You can see here, at the moment, we have a repetitive cell barcode for the same antigen species and the same antigen gene. If we want to plot the antigen epitope on the UMAP cell canisters, we need to break down the annotation into a single value per cell. Then we can visualize the antigen epitope on the UMAP cell canisters. For example, here we have 3 CMV antigen species and they have the same cell barcode, so we can just keep 1 for the UMAP visualization. So now we can break down the annotation. Let's do it now. So after that, we can visualize the antigen epitope on the cell canisters. We can use the plot embedding function, and we have the cell canisters in the single-cell RNA sequencing data for this dataset. So let's plot the antigen species on the cell canisters. If we run, you can see we generated a UMAP to show the antigen species on different cell canisters. You can see in total in this dataset, we identified the antigen epitopes for 12 different antigen species. Okay, that's today's demonstration. I showed you how to load the public available database and annotate the antigen epitope in this demonstration dataset. And also we can plot the antigen species using the cell canisters from the single-cell RNA sequencing dataset. So I'm going to stop from here. I hope to see you in my next video tutorial.